This lecture is on prokaryotic diversity. It's chapter 4. The first thing we're going to talk about is prokaryotic habitats and functions. So prokaryotic habitats are really everywhere. Prokaryotes are everywhere. Whether you're a bacteria, a fungi, an archaea, a protist, they are all across the globe. Forests, in oceans, in the desert, in the tundra, everywhere. In land, water, and air, even in very extreme environments, like environments that are very hot, under a lot of ice, under extreme pressure, high salinity, high sulfur, lots of different things. There's also a great deal of prokaryotes in and on the human body. There are more bacterial cells than human cells in and on a human, especially on the mouth, nasal cavity, throat, ears, GI tract, genitals, and skin. There are many, many microbes in soil, up to 10 billion cells of bacteria per gram of soil, which really is not a lot of soil. Soil is very, very rich in microbes. Microbes are really, really important for soil, for the breakdown of organic matter, for taking nutrients and turning them into things that plants can use. That's called fixation, such as the fixation of nitrogen. They are also very good at cleaning up the environment. They can degrade toxic chemicals and oil spills. An interesting thing I saw a little while ago was that prokaryotes actually may be contributing to climate change because of carbon thawing from the permafrost. By the melting of the ice caps, that is being metabolized by prokaryotes, making more carbon dioxide and methane, which are greenhouse gases. So greenhouse effect leads to global warming, leads to melting ice caps, leads to more global warming. Let's talk a little bit about fixation because it's such an important thing. Some elements like nitrogen can't be used effectively by plants as they exist in the environment. So microbes, like some bacteria and some fungi, take up elements like nitrogen and turn them into different molecules that the plants can then take up and use. So a great example are the rhizobium bacteria. They take nitrogen from the air, which is N2, and they turn it into ammonia through a chemical reaction. And the plants can actually take up the ammonium much better than they can take up the nitrogen. And then so that incorporates the nitrogen into the food chain because then the plants have the nitrogen in them. Nitrogen is very important for things like amino acids and nucleotides, so very important building block in our cells. And then the animals will eat them and so on and so forth, and it goes up the food chain from there. All right, so here's an overall diagram of how nitrogen is used in the environment and different organisms that help put that nitrogen into the food chain. So a large focus in microbiology is on pathogenic microbes, which makes sense if you think about it because pathogenic microbes are very, very important to us because they can seriously disrupt our way of life. The good news is that less than 1% of bacteria are human pathogens and no archaea are human pathogens. In addition to being pathogens, bacteria can contaminate food, causing food spoilage or food poisoning, and infection and disease when they enter the body. There are lots of microbes everywhere around us. I found this was really interesting. There are a lot of microbes in the air, and we know that now more than ever right now with COVID-19 going on. But in the air, even around in your home, there are thousands of different kinds of microbes. The good news is most of them won't make you sick. Something that's really unique that microbes can do that help give them a survival ability is that they can be metabolically flexible, right? They can switch from one energy source to another, such as facultative anaerobic bacteria that can metabolize in the presence of oxygen or not. They can change their food source from carbon dioxide to carbon being taken up or things like that. Another example is cyanobacteria. So cyanobacteria are bacteria that, like a plant cell, can photosynthesize. But if there's not enough sunlight, they can actually switch from photosynthesis to lipid metabolism, so breaking down fats for energy if there's not enough sunlight around. Let's talk a little bit about symbiotic relationships, since a lot of relationships with microbes are symbiotic. Prokaryotes live in communities. A community is a group of interacting populations, okay? A population is a group of organisms belonging to the same species. So population, same species, community, lots of populations living together. There's a whole field of microbiology called microbial ecology. And what that does is it studies the association between microbes and their environment. When we're talking about interactions between microbes and their environment, we're talking about symbioses. Okay? And there are lots of different kinds, like mutualism, commensalism, parasitism. You've probably heard of those before. There's also amensalism and neutralism. In mutualism, both organisms benefit from the shared environment. So a couple of examples. E. coli in our gut makes vitamin K for us, which we don't make ourselves and don't get a lot of from our diet, unfortunately. But vitamin K is really important in your blood clotting properly. Bacteroides, bacteria, can digest plant material that we otherwise can't break down, and they live in our gut. So we get a benefit from that because they break down material for us so that we can get nutrients out of it, and they also get nutrients from breaking down the material as well. In amensalism, one organism is harmed, but the other is unaffected, so it doesn't get a benefit at all. For example, Propionibacterium lives on your skin. It can cause acne, and it produces bacteriocins, which stop the growth of other closely related bacteria. Okay, so Propionibacterium is harming other closely related species of bacteria because they are secreting bacteriocins. In commensalism, one benefits and the other is unaffected. Like Staph epidermidis. Staphylococcus epidermidis is a bacteria that lives on the skin. It's related to Staph aureus and MRSA, but it's a different species of Staphylococcus. It uses dead skin cells as food, but it does not help or harm us. So it lives on our skin, like as kind of like a free ride, really. But that's commensalism. In neutralism, both neither benefit nor are harmed, and this one's a little complex. Biologically active bacteria can coexist without affecting each other. In neutralism, both of the organisms neither benefit nor are harmed. So they essentially just coexist without affecting each other at all. So they're neutral. For example, Bacillus anthracis and its endospores can coexist without affecting each other. And then the one we're probably most familiar with is parasitism, right? This is all of our pathogenic microbes, right? Disease-causing bacteria, one benefits, the other is harmed. One that benefits is the microbe, and we're the one that's harmed usually. Here's a great gram stain of Clostridium difficile, C. diff. C. diff are gram-positive rods, so dark purple rod-shaped bacteria in a stool sample, which has some little gram-negative bacteria, but also these giant pink globs. These are human cells, or remainders of human cells, or other eukaryotes, because they're so big, if you will, and they're gram-negative. Because cells that don't have any peptidoglycan, so non-bacteria, will stain gram-negative in a gram stain just because of the safranin dye. All right, so we wouldn't be doing justice to microbial communities if we didn't talk about our microbiome. Our microbiome is so important. It's all of the organisms living in a certain environment, like the human microbiome in our bodies. So in our microbiome, we have two kinds of microbes. We have resident microbes that are always there, and transient microbes that can come and go. Our human microbiota is very, very diverse. There are different populations of microbes in different areas of the body. So the skin, the digestive tract, the mouth, the urogenital microbiome, all of those are very unique. Something that's really interesting is that newborn babies are first exposed to microbes during birth. And this starts the establishment of the microbiome, especially lactobacillus bacteria that lives in the gut. So babies born by vaginal birth have a lot more exposure to microbes than babies born by cesarean. However, babies born by cesarean will still develop their microbiome, but it might be a little bit slower, or the populations of microbes that it encounters first might be a little bit different, like skin microbes versus urogenital microbes. And after birth, infants get their microbiome from everything they interact with. Parents, other care providers, anyone or anything that comes into contact with the baby can help establish that microbiome, which can be good or bad. Microbiota are super important for our human health. Think of this as our good bacteria, right? Our good bacteria takes up space and uses nutrients that might otherwise be used by pathogens. So because the good bacteria are there, the bad bacteria don't have enough space or nutrients to grow. Great example of that, lactobacillus is really common in the female genital tract. It reduces the pH and inhibits the growth of pathogenic yeasts, right? But if you kill off that bacteria, then you're going to have the overgrowth of yeast, right? So how do we know that a species is a species? And the rest of this chapter is really going to focus on classifying microbes, and prokaryotes specifically. So in animals, we classify a species as a group of organisms that are very similar and interbreed with one another. Bacteria don't really interbreed, besides conjugation and transduction and those things. So we have to use characteristics that are a little bit different to categorize them. So we use morphological features like shapes, staining patterns, biochemical and physical differences, and now nowadays genetic analysis to help determine what is a species. Back in 2008, the National Institute of Health started what was called the Human Microbiome Project. People were starting to really be aware that the microbiome existed, and we really didn't know a lot about it. And so scientists started to study the microbiome in depth and discover kind of what microbes were present where and when. The goal of that was to help us understand the relationship between human microbes and disease. A couple of drawbacks to this are that some microbes can't be cultured, so we don't know that they exist and they can't be studied. However, if we're looking at genetics from an entire biome, that will help us get all of the genetic material that is in that microbiome so that we can try to figure out what species are there. Something interesting is we all have some pretty serious microbial pathogens in our microbiota, but the other microbes in our immune system are able to keep those at bay, usually. So we're going to talk a little bit about prokaryotic taxonomy. So how prokaryotes are categorized and where they fall in the tree of life. So some interesting things to note. There is a really special group of bacteria called the deeply branching bacteria, and they are over here where this little star is. Okay, so they are one of the first kinds of bacteria to evolve on their own. They live in hot, acidic, UV exposed, or anaerobic conditions. And what's interesting about that is those are the conditions that were present on Earth before other life on Earth existed. But for the most part, we're going to break the bacteria up into gram-negative and gram-positive bacteria. So gram-negative bacteria are broken into a few groups: the proteobacteria, CFBs, and the spirochetes. The gram-positive bacteria are broken down into two groups based on what their nucleotides are like. So if they have more C's and G's than A's and T's in their DNA, they are in the high G plus C. Or if they have less, they're in the low G plus C. We'll talk more about that in a minute. All right, so here is a dichotomous key of prokaryotes. So what a dichotomous key is it is kind of like a tree, if you will, a decision tree. You start at the top with all of the things, and then you keep splitting them into groups until you end at one specific thing. So if we take all the prokaryotes, we can split them into bacteria and archaea based on whether they have peptidoglycan or not. Okay. And then we can take our bacteria, we can break them into gram-negative and gram-positive. We can break our gram-negatives down further and our gram-positives down further. So you start big, you keep breaking it into smaller groups based on shared characteristics. So we're going to talk about the gram-negatives first. We're going to talk about the proteobacteria. Okay, so for this again, gram-negative, we have proteobacteria and non-proteobacteria. We're going to start with proteo. The proteobacteria are defined as purple bacteria and their relatives. There are 5 different divisions of proteobacteria, and each of them have a Greek name: alpha, beta, gamma, delta, and epsilon. And talk a little bit about what are in each of these groups. The alpha proteobacteria are able to live in really low nutrient environments. The betas are kind of opposite in that they require a lot of organic nutrients. They usually grow in environments like intestines where there's a lot of nutrients passing through. And we have the gamma proteobacteria, which is probably the largest group of bacteria and gram-negative bacteria that exist. Many pathogens are here, and as well as many other bacteria. We have our delta proteobacteria, which are bacteria that live usually in high sulfur environments. And then we have our epsilon proteobacteria, which consist of our microaerophiles. A couple of medically relevant ones here are Campylobacter and Helicobacter. H. pylori can cause stomach ulcers and stomach cancer. So here are the alpha proteobacteria. Here's a dichotomous key of how they can be broken down. So do they cause disease? No. Yes. Are they obligate intracellular parasites? Do they absolutely need to be inside a cell to cause disease? Yes. No. So you can keep breaking them down into groups until you arrive at one specific bacteria. So if you are an alpha proteobacteria, you cause disease in humans, you're an obligate intracellular parasite, and you cause spotted fevers, you are in the Rickettsia genus. Okay. The betas here are a very diverse group, so this tree looks a little bit different. It can oxidize sulfur or form sheaths, or they're foci shaped or helical or rod shaped. If you're a rod, do you cause pertussis? Well, then you're Bordetella pertussis, which is the causative agent for whooping cough and other Bordetella bacteria. Are you found in sewage? Are you not? So beta proteobacteria is a very diverse group. The gamma proteobacteria, remember this is kind of our biggest group of gram-negatives, okay? And they're very diverse. You're not going to need to know all of these divisions and breakdowns. The delta proteobacteria are a very small group, but very interesting and unique. The desulfovibrios are sulfur bacteria. Mycococcus bacteria, those are really interesting. We'll talk more about them later. Then we have the epsilons, which are really just Campylobacter and Helicobacter. All right, so that's proteobacteria. Then we have our non-proteobacteria. Our non-proteobacteria include the spirochetes, the CFB group, which stands for Cytophaga, Fusobacteria, and Bacteroides, and then the Plantomyces. Spirochetes, remember, are long, spiral-shaped bacteria They are flexible and they are very motile because they have an axial filament, which remember is that flagella that's kind of embedded in the outer membrane of the cell, which makes it spin like a corkscrew through its environment. Borrelia burgdorferi, which causes Lyme disease, is a great example of a spirochete, as well as Treponema pallidum, which causes syphilis. In the CFB group, it's a very diverse group, but they're similar in their DNA sequences, so we know that they are more related. They're all rod-shaped, and they live in anaerobic environments. The cytophaga bacteria are motile, aquatic bacteria that glide through their environments. The fusobacteria are commonly in the human mouth, and they can cause severe infections and diseases like gingivitis and periodontitis. And the bacteroides are bacteria that are actually really good for our gut. So a very diverse group, but very similar in their DNA. So they're highly related, but very different in their characteristics. And then we have our plantomyces, which are aquatic bacteria. They reproduce by budding, or they can make swarmer cells, which we'll look at a little while later. So the phototrophic bacteria are a group of bacteria that aren't an official taxonomic group. They're kind of like an unofficial category because they're so similar. They use sunlight to make their energy like a plant, but they are not photosynthesizers in the sense that they use green pigments like chlorophyll and make oxygen. So most of these phototrophs do not use or produce oxygen. They are the purple bacteria and the green bacteria, and there's a picture of them here. Then we have our cyanobacteria. So cyanobacteria are aquatic. They can also live in soil and rock environments, because remember, soil is very diverse and also fairly moist. They live at a range of temperatures. These guys make oxygen for us during photosynthesis. They are very prominent oxygen producers on our planet. They can live unicellularly or in colonies or other formats. They have organelles that are kind of similar to chloroplasts, but not quite chloroplasts. And sometimes when they overgrow very rapidly in small bodies of water, they can cause cyanobacterial blooms with lots of toxins. So occasionally in the summer, you'll hear about an algal bloom or a cyanobacteria bloom in like a lake or things, and then you have to avoid that area for a while. So that's one of the reasons why. All right, next we're going to talk about our gram-positives. The gram-positives are divided up based on their DNA, if they have more G's and C's or A's and T's. If they have more G's and C's, then they're the high G plus C group. These guys are also called actinobacteria, lots of naturally occurring bacteria in the environment. They can have lots of different kinds of peptidoglycan in their cell walls. They're usually small rods or cocobacilli, which are kind of a cross between a coccus and a bacillus cell. And they often are filamentous. Most of them live in the soil. Most of them are aerobic. Actinomyces is a genus of soil bacteria, but it's also an opportunistic pathogen of the gums and mouth. Another example are the mycobacteria. Those are bacteria that we can't really gram stain very well because they're covered in that mycolic acid. The low G plus Cs are a very diverse group. There's a lot of pathogens in this group. The Clostridium right here, there's lots of different Clostridium, not just C. diff, but most of them are anaerobes that make endospores that can be pathogenic. So because Clostridium difficile makes endospores, that is one of the primary reasons it is so hard to get rid of, because those endospores can live on surfaces for a very, very long time. So you have to really disinfect and wash hands and everything to stop the spread of C. diff. Other bacteria here are Streptococcus, Lactobacillus, Enterococcus, Bacillus. It's kind of a laundry list of gram-positive pathogens. Let's talk a little bit about Streptococcus, and we'll come back to this when we talk about human pathogens later on. But there are lots of kinds of Streptococcus, and they are all pretty much human pathogens. They are chains of spherical cells. Remember, strepto means chain and cocci is spherical cells. They're classified based on whether or not they can lyse red blood cells. If they can lyse red blood cells, then they're called hemolytic. So you might have heard of things like beta hemolytic strep or things like that. So that's where those words come from. A great example of a strep species is Streptococcus pneumoniae. It exists as diplococcus and can cause pneumonia, but it can also cause other infections like meningitis, osteomyelitis, endocarditis, and even septicemia. Here's a couple pictures of some strep. Here is a gram stain of Streptococcus pyogenes. So gram positive, dark purple. Streptococcus chains of circular shaped cells, which are kind of hard to see here, but they are circular. That's looking at a gram stain on a microscope. This is looking at colonies of the bacteria on a blood agar plate. So this plate has the nutrients in the agar that we would normally have in a plate, but also blood, so that you can see where if the bacteria can break down red blood cells. All right, in this group, we also have our bacillus species. There are many, many different species of bacillus bacteria. Most of them can make endospores. They're either aerobes or facultative anaerobes. They're used for lots of different things. We can use them to make things like antibiotics. We can use them to produce enzymes that we can then harvest and use for things like detergents. A few example species here are Bacillus anthracis, which causes anthrax, Bacillus cereus, which can cause food poisoning, but it's also a soil-dwelling bacteria. Staphylococcus also belongs to this group, interestingly enough, although the morphology is completely different. They are related through their genomes. And also Mycoplasma belong to this group, which don't really have cell walls because they're such small bacteria. And then let's go back to our deeply branching bacteria. So they are thought to be the first bacteria that ever evolved from the universal common ancestor, from whatever we all started out as. They're essentially the first bacteria. They likely still exist today, although if they do, we're having a lot of trouble finding them. They lived at high temperatures because the earth was a lot hotter back then. So we can find some of their relatives in places like hot springs or hydrothermal vents deep in the ocean. Some of them are very, very resistant to UV light. When the earth was first formed, we didn't really have the good atmosphere that we have today. Our atmosphere is able to remove a lot of the harmful UV rays from the sun, so that in general, life on Earth isn't really affected by it. But it didn't always be that way. And so the very first bacteria were much more resistant to UV light than bacteria are nowadays. Or resistant to radiation. A really interesting species of bacteria called Deinococcus radiodurans has been found at places like Chernobyl and Fukushima, where there have been really high amounts of radioactivity present. And they're really resistant to any effects by radiation, because radiation would normally cause mutations in the DNA. In Deinococcus radiodurans, not only are they resistant, but they can repair those mutations when they occur. So lots of really cool research being done on Deinococcus radiodurans. And finally, we have our archaea. We don't get to talk a lot about archaea. The reason for that is we don't know a lot about them. They're very, very diverse. They can be found in any habitat, but most of them, because their environments are so extreme, they're very difficult to culture in a lab. There are at least 5 phyla of archaea, but most of them are still undiscovered. So those 5 phyla are listed here, and with some characteristics about their environment. So for example, high methane, halophiles like a lot of salt, hyperthermophiles, they get very, very hot. Some bacteria live in high nitrogen environments, things like that. or some archaea rather, not bacteria. And that's the end. Thank you.